who's so Amanda is an assistant professor in the Department of Molecular and Cellular Biology at Harvard University. And today she's going to be talking about the regulation of imprinted gene expression in the brain. So I'll hand over to Amanda.
Great. Thank you so much. It's really my pleasure to be here today and to have this opportunity to share with you all and introduce you to some of the central questions that we're asking in the lab, and to tell you about some recent work that we're doing within the context of genomic imprinting. So just by way of introduction, genomic imprinting is an epigenetic process that ultimately results in genes that are predominantly expressed from only one of the two parental copies. So I've illustrated this here as a gene predominantly expressed from the paternal copy, or this nearby maternally expressed gene. And although this is a simplified schematic, imprinted genes do typically occur in clusters in the genome. These clusters contain both paternally as well as maternally expressed genes. And the imprinted expression of all genes within the cluster is controlled by differential DNA methylation that occurs at these discrete regions of DNA known as imprint control regions, or ICRs, where one parental copy is methylated and the other is not. These differences in DNA methylation are inherited directly from the gametes, such that following fertilization and throughout each cell division and development, the differential methylation at these regions is rather remarkably maintained. Imprinted genes are expressed across many different tissues, and historically, the functions of imprinted genes have been best studied in the placenta and early embryo. But in more recent years, it's been shown that imprinted genes are highly expressed within the brain. And here they play a variety of roles in regulating different aspects of brain development and ultimately affecting the behavior of the organism. The importance of imprinted genes in the brain is further exemplified by a number of human imprinted disorders that have neurological phenotypes. So I'm going to highlight in just a little bit more detail one of these imprinted disorders called Burke-Barrell syndrome. So Burke-Barrell syndrome arises from mutations in a particular imprinted gene that is exclusively expressed from the maternally inherited copy. This gene encodes a potassium leak channel called KCNK9 that's expressed in neurons and is important for membrane potential. So individuals who inherit certain mutations in the KCNK9 gene, specifically from their mother, develop Burke-Barrell syndrome as a result of this mutant KCNK9 protein that's expressed within their neurons. So as a general principle in terms of the therapeutics of treating different imprinted disorders, one goal is frequently to perhaps try to reactivate the expression from the other parental allele, in this case, the paternal allele, which is usually intact within patients. But this actually requires us to first understand how the paternal copy of this gene gets silenced, and then find some way to intervene and disrupt that process. And so for KCNK9 specifically, it's not immediately obvious how the paternal copy of this gene gets silenced. But in other scenarios, like in the case of Angelman syndrome, this type of principle of drug design has been quite effective. So just to briefly walk you through this, Angelman syndrome is also caused by aberrations in the maternal copy of the genome. In this case, this affects a particular maternally expressed gene that encodes a ubiquitin ligase called UBE3A. And if you inherit deletions of this gene from your mother, you have a complete absence now of the UBE3A protein within your neurons, and that's what results in this neurodevelopmental disorder. So it was quite exciting when Art Boudette's lab discovered that the paternal copy of this gene, which is intact in most patients, but silenced by genomic imprinting, is actually silenced by the presence of this cis-repressive long non-coding RNA. And so with this information, then we reasoned that perhaps we could develop a therapy for Angelman syndrome by knocking down this repressive long non-coding RNA, thereby reactivating the intact paternal allele within patients. So this is what we were able to do using antisense oligonucleotides in cultured neurons or a mouse model of Angelman syndrome. And this works quite well to actually restore UBE3A protein levels in neurons. And this is now in clinical trial. So I'm using this as an example of this fundamental principle that by better understanding how these imprinted regions are controlled, this could highlight particular therapeutic avenues in disease. So I wanted to just introduce you to some of the overarching research questions that we're asking within my lab. First is that we're asking questions about the basic molecular mechanisms that give rise to imprinted gene expression in different tissues like the brain. Second, we ask questions about the functions of imprinted genes. What are these genes actually doing within the brain? With the hope that over time we may be able to gain a better understanding of why these genes have evolved imprinted expression. And here we have a particular interest in the large number of non-coding RNAs that are imprinted in the brain. I've already alluded to an example of an imprinted long non-coding RNA, but there are also many small non-coding RNAs like microRNAs and SNO-RNAs that are imprinted in the brain that we're interested in understanding the functions of. And ultimately, we hope to take what we learn about how these genes are regulated and what they're doing to think about novel therapeutic strategies for different imprinted disorders. So for the talk today, I'm going to focus on some recent work that we've done in which we're characterizing the basic mechanisms controlling imprinted gene expression. So we know that the imprinted expression of a gene is ultimately controlled by differential DNA methylation at these imprint control regions. If you manipulate methylation in these regions, or for example, delete the ICR, then you lose all imprinted expression from the locus. So genes either become biallelically silenced or biallelically expressed. But there are still open questions that remain about how exactly these ICRs work. First, it's not clear how these ICRs have apparent long-range activity in being able to regulate the imprinted expression of genes that are located far away, meaning tens to hundreds of kilobases away within the genome. Second is that although DNA methylation is incredibly stable across different tissues throughout development, the imprinted expression of genes is actually surprisingly dynamic. So you can have a gene that is not imprinted, it's biallelically expressed in peripheral tissues, but then exhibits imprinted expression specifically within the brain. So although imprinted expression is controlled by DNA methylation, it seems like there are additional factors that are at play. And one layer of gene regulation that we've become very interested in recently is the potential for chromatin structure to impact imprinted gene expression. So although we typically think about DNA as just this linear sequence, DNA is actually highly folded within the cell to specific 3D conformations. And this is done with help from factors like CTCF and cohesin. And the way in which DNA is folded within the cell ultimately helps determine which genes get expressed. One layer of this structural organization comes in the form of topologically associated domains, or TADs. You can think about these as gene neighborhoods in which regulatory elements that live in the same neighborhood have a much higher frequency of interaction and regulate one another, and they're insulated from interacting with regulatory elements that live in other neighborhoods or TADs. So recently we've begun to wonder if perhaps maternal and paternal DNA are folded differently from each other within the cell. And if so, if these differences in their folding may actually give rise to differences in gene expression between the two parental alleles. So I hope you'll bear with me as I chose to talk about a rather DNA-centric project at this RNA forum. But what we're ultimately aiming to understand here is how the cell decides if an RNA gets expressed from one parental copy versus the other. And this layer of gene expression regulation hopefully is applicable to many of you, even outside of the context of genomic imprinting. So there's precedent for thinking that there could be folding differences between the two parental copies. So not only do we inherit differences in DNA methylation from our parents, oocyte and sperm actually have very dramatic differences in their chromatin structure due to the differences in how DNA is packaged in these nuclei of very different sizes. Second is that one of the key factors involved in forming chromatin contacts, called CTCF, binds to DNA in a methylation-sensitive manner. And it's been shown at a number of ICRs that CTCF is bound to the unmethylated parental allele, but not to the methylated allele. There's evidence for these structural differences between the parental genomes from recent Hi-C studies, as well as a number of studies that have occurred on the IGF-2 imprinted locus over many years. But we wanted to obtain a really high resolution readout of this chromatin structure in a system where we could distinguish maternal and paternal DNA. So a postdoc in the lab, Min, performed region capture Hi-C from hybrid mouse brain tissue. So in these experiments, we crossed two different strains of mice such that the resulting F1 progeny have a high density of single nucleotide polymorphisms that we can use in sequencing-based approaches to distinguish maternal or paternal features of the genome. We then isolated the cortical tissue from these mice and performed region capture Hi-C for some of the major imprinted domains in the genome. In order to distinguish imprinting or parent of origin effects from strain-specific or SNP effects, we also perform these experiments in the reciprocal hybrid cross. So just for those of you who may be unfamiliar with Hi-C, I wanted to go over just some of the key basis of this particular assay and approach. So here we have a cell with chromatin that's folded in a particular orientation. You can pay particular attention to these colored regions of the genome, which are far apart in the linear sequence of the DNA, but are brought into close spatial proximity within the cell. So in these experiments, you cross-link your cells, digest the DNA with restriction enzymes, and then ligate together proximal pieces of DNA that you can then purify. And from this, you make high throughput sequencing libraries, where the key thing to note is that pieces of DNA that are spatially located together within the cell can now be captured in a single sequencing read. And you can use these sequencing reads to then map them back to the linear sequence of the genome and use this information to calculate the interaction frequencies between any two pieces of DNA, such that DNA that's folded into a TAD results in this sort of triangle-like appearance on these Hi-C interaction maps. So we performed these experiments and then used biotinylated probes to enrich for imprinted regions of the genome that allowed us to generate high resolution maps of the chromatin structure across these imprinted domains. For the talk today, I'm just going to focus on the results of one particular imprinted region. This is an imprinted region around the KCNK9 gene, which is the gene that I introduced earlier that's implicated in Burke-Barrell syndrome. And we're able to use the SNPs within these Hi-C reads to look at either the maternal DNA structure, which is shown on the top in red, or the paternal DNA structure shown on the bottom. So there are many imprinted genes within this imprinted cluster that surround this central region of differential DNA methylation. And what we observe is that on the maternal allele, there's one large TAD that forms that stretches across most of this imprinted domain. In contrast, on the paternal allele, this is now actually broken up into the formation of two smaller TADs that are anchored by CTCF binding to the unmethylated ICR. So if I were to draw this in a cartoon format, it would look something like this, where there are these very clear structural differences in how these parental genomes are folded across this imprinted domain. So this data, along with other imprinted domains that we have looked at, have allowed us to conclude that the chromatin structure is indeed itself imprinted. But we then wanted to ask the question, does it matter? Does it matter that the DNA we get from our mom is folded differently in our cells than the DNA we get from our dad? And more specifically, do these differences in chromatin structure actually impact the genes that are being expressed from each of the parental copies? So to answer this question, a graduate student in the lab, Daniel Loftus, turned to an in vitro neuron differentiation system in which we can perform perturbation experiments more readily. So in this system, we have hybrid mouse embryonic stem cells that were able to differentiate in culture by neurogenin 2 induction to what I'll refer to as induced neurons. And first, he repeated the region capture Hi-C within this system to show that the overall chromatin structure that we observed in the brain is also present within the induced neurons. So once again, just to reiterate that on the maternal allele, there's one large TAD that forms across this imprinted domain, whereas on the paternal allele, we now see the presence of these two paternal specific subtads within this region. So Daniel then wanted to try and find a way to ablate the imprinted chromatin structure to ask what effect that has on the imprinted expression of genes from the locus. And he did this by performing a CRISPR-mediated deletion of the two CTCF motifs that are present within the ICR on the paternal allele. So overall, in these CTCF deletion cells, most of the ICR has been left intact, but there are these two small deletions that delete the CTCF binding sites. And when he does this, this results in a complete loss of the parent-specific chromatin structure within these cells. So now on the paternal allele, those two previous paternal-specific subtads have been lost, and the paternal allele now has an overall folding or conformation that looks very similar to what occurs on the maternal allele. So now within these cells, he's able to ask, once you lose imprinted chromatin structure, what happens to imprinted gene expression? So here we're performing droplet digital PCR as a way of being able to quantify the allelic biases in gene expression. So just to orient you how these plots look, biallelic expression or equal expression from both parental copies is seen here at y equals 0. Positive values indicate maternal biases in expression, and negative values indicate paternal biases in expression. So you can see that in the wild type cells, KC and K9 and this PEG13 long non-coding RNA have very strong imprinted expression. There's much more subtle levels of imprinted expression of these more distal genes, TRPC9 and AGO2. But in all cases, when we ablate the imprinted chromatin structure in the deletion cells, we also lose imprinted expression. So all the genes trend towards this biallelic y equals 0 value. So what this data shows is that it seems that the imprinted chromatin structure is important for informing the imprinted expression from the locus. So we then wanted to ask, well, how exactly does that happen? How does the chromatin structure, why does the chromatin structure matter? And so to address this, we turned our attention to one of the key genes within this region, KC and K9. And what Daniel observed is that there's a potential enhancer within this region based on brain ChIP-seq data sets. This enhancer has high levels of H3K27 acetylation and H3K4 monomethylation. And importantly, this enhancer lies in two different TADs between the parental alleles, where he hypothesized that there could be differences in the enhancer-promoter interaction that are actually driving this maternal specific expression of KC and K9, And on the paternal allele, the enhancer would now be insulated against interactions with the KCNK9 promoter due to the structural differences between the alleles. So in order to test this hypothesis, he turned to the mouse embryonic stem cells. And first he showed that within the stem cells, the overall imprinted chromatin structure is already present. So it looks very similar to what we've observed in brain and neuron tissue. But KCNK9 is not yet expressed, nor are there any active marks at the enhancer. So this gave him a system then where he could artificially induce activation of this distal enhancer and ask what happens to KCNK9 expression. And it's important to note that in this system, we're actually driving biallelic activation of the enhancer and then asking what happens to the imprinted expression of KCNK9. So what he found is that when he forces the transcriptional activation of this enhancer in embryonic stem cells, he upregulates KCNK9 about 100-fold. But importantly, when he then asked from which allele does KCNK9 get expressed from, he observed this preference in KCNK9 induction from the maternal allele. As a control, he also drove the transcriptional activation directly from the KCNK9 promoter. And when you do this, you can achieve a very robust upregulation of KCNK9, but this occurs in a biallelic fashion, indicating that both the maternal and paternal promoter are capable of being expressed within the cells, but that the activation of this distal enhancer results in a preferential expression from the maternal allele due to these structural differences. In order to further test this, he then repeated this experiment in the cell line where he had a loss of imprinted chromatin structure. So this is done in these cells where he deleted the CTCF binding site and the ICR of the paternal allele. So now there's no imprinted chromatin structure present, and when he drives the activation of this distal enhancer, he's still able to activate the expression of KCNK9, but now this occurs in a perfectly biallelic fashion, indicating that indeed the structural differences within this region are really promoting the imprinted expression of KCNK9. So just to sort of zoom out in the conclusion of this talk, I want to go back to something that I said in the introduction, which is that there are strong structural differences in the folding of DNA between oocyte and sperm. So what exactly happens to the chromatin structure then following fertilization? So following fertilization, there is a pretty strong erasure of chromatin structure, particularly on the paternal allele. But then gradually but rapidly throughout these early cell divisions in development, the chromatin structure is reestablished. And overall, the structure of both parental genomes converge on a common structure. But what I told you today is that if we zoom in on particular regions of the genome, these imprinted regions, we can observe the continued presence of structural differences between the parental genomes. And at least in the case of the KCNK9 region, we think these structural differences matter, and that alternative enhancer promoter interactions that are occurring between the parental alleles are thereby driving parent-specific gene expression. So with that, I want to thank the individuals who have really contributed to these studies. The work that I was presented today was most directly done by Min and Daniel, and I'd like to thank all of the funding sources for supporting this work, and I'm happy to take any questions.
Thanks, Amanda. Very interesting. Beautiful data. It's one of those situations where you don't even need a model. You can just look at the Hi-C and it's already quite revealing, I think. So I think we have a question in the box. Let's see. So, yeah. Thank you for your great presentation. A little bit out of scope of the experiments you showed, but is there any indication that the metabolic status of the cells may change the imprinting status of these genes? This is from Nick Sotakis.
Yeah, I mean, it's an interesting question. So there's a lot of links between genomic imprinting and different aspects of metabolism. So a lot of this relates to the studies that have been done on genomic imprinting in the context of early development and the placenta and the growth of the embryo within the mother. And it sort of relates to maybe why we've evolved imprinted expression of particular genes. So many imprinted genes are themselves involved in different aspects of growth regulation and metabolism. The exact relevance or extent to which that applies to imprinted gene expression that we see in the brain is not as clear. And IGF-2 is actually a really interesting example of this. IGF-2 is probably classically one of the most studied imprinted genes. And in the embryo, it's expressed from the paternal allele and is acting to drive the growth of the embryo. Interestingly, IGF-2 actually switches to maternal specific expression in the brain, and that kind of regulation hasn't really been studied. So within the context of these particular experiments, we haven't looked at any sort of metabolic status of the cells or tissues that we've manipulated, but there is precedent and reason for thinking about some of these questions.
Okay, thanks. So I'm just wondering, so you have a nice system where you have an enhancer that's working within this loop. And obviously there's a lot of chromatin focused, but enhancer RNAs exist, right? So I'm wondering if this could be a nice system to explore whether you need the RNA or whether it's simply, the transcription factor binding to the element that's more important.
Yeah, it's interesting. I mean, so there is when you look at the ChIP-seq data for the active enhancer marks in brain data, most data sets are not allelic, so you can't really distinguish maternal versus paternal features in most of the existing data. But there is one allelic H3K27 acetylation data, which actually seems to indicate that there might be some sort of maternal bias in the enhancer activity of the locus that I showed. So in terms of looking at like enhancer RNAs, it would be really interesting to see actually if there are sort of allelic enhancers that could really be sort of driving some of this allelic gene expression, but we don't know the answer to that yet.
Thanks. There's another question in the Q&A box. This is from Wen Tang and says that they might have missed this. Why is imprinted chromatin structure established only in brain or neurons?
Yeah, so actually it's a little bit of a correction there. I didn't show the data, but the imprinted chromatin structure is actually already present when we've looked in embryonic stem cells. So in some regard, maybe this seems like a little bit of the misfit data. I'm saying that the imprinted chromatin structure is what's driving imprinted gene expression. There's actually no imprinted expression of this locus in embryonic stem cells, yet there's imprinted chromatin structure. So this is data beyond what I've shown here. But what we think is happening is that the imprinted chromatin structure, which is established very early in development, we believe as a direct result of differential CTCF, which is bound to these differentially methylated regions, is sort of setting up or poising the locus for the ability to gain imprinted expression in tissues where these enhancers become activated, like the brain. So actually what we see is that the imprinted chromatin structure, at least for this locus where we've looked, seems quite stable. It's relatively uniform across different cell types or tissues, independent of the imprinted expression from the locus. But in tissues where you have this brain-specific enhancer activation, you've now sort of poised the system in a way to give rise to imprinted expression. And that's how we're thinking about it.
Thank you. So we have another question from Guaokei Zhong. So their question is about how is this initial chromatin, either paternal chromatin or maternal chromatin, established in the first place?
Yeah, so it seems like all the existing data aligns with a model in which so differential DNA methylation is, ultimately the mark, the primary mark that's controlling genomic imprinting. It is the imprint mark. So as I said, that's actually established either during oogenesis or spermatogenesis, so during formation of the gametes. And then following fertilization, those methylation differences are maintained. And many of these ICRs have binding sites for CTCF, which is one of this sort of key regulators in the chromatin structure formation. So essentially, the differences in DNA methylation are leading to differences in CTCF binding to the parental genomes. And that thereby is setting up a system in which you can have structural differences between the two parental genomes. So yeah, it's not a random process, but it's happening as a result of this downstream methylation.
Okay. Well, I'm just checking if there are any more questions, and it doesn't appear to be the case. Oh, wait, there is one. One last question here. So, enhancer and KCNK9 are still close to each other, even in the insulated state. Do you somehow measure the DNA distances in parental and maternal states? Or sorry, did you somehow measure the distance? So I guess the point is, even if it's in a separate loop, they're still kind of close together. So could you talk something about relative distance between the enhancer and the promoter and why it can't work when it's in a proximal loop?
Yeah, I mean, I think the fact that they are sequestered into different TADs really does make a difference there. So the enhancer is actually even closer to some other gene promoters than it is to the KCNK9 promoter. Yet when you activate the enhancer, it's preferentially activating the expression of KCNK9 when it's in the same TAD together. So I mean, I agree with you that the enhancer and KCNK9 are still close to each other, even on the paternal allele, but the fact that they're sort of isolated into two different TADs and that CTCF has insulating roles in between them, that seems to be protecting, to some extent, this enhancer-promoter interaction.
Okay, well, that's great. And just perfectly on time. So I wanted to thank both speakers for excellent talks and the audience for their questions. And with that, I will conclude this RNA Collaborative Seminar Series. I believe the next one is October 11. So tune in next... oh, sorry, that's already passed. I don't know when the next one is. But anyway, so that's the end of this one. So thank you. Good night.